Hi everyone. In the CPGET Microbiology Important MCQ, in this video, the series 13 is going to be discussed with the 25 important questions. Majorly, I am covering the molecular biology in this video. Let's begin. On glycolysis, ATP is formed by options are A. Oxidative phosphorylation, B. Photophosphorylation, C. Substrate level phosphorylation, D. All of the above. And the answer is going to be the substrate level phosphorylation. In glycolysis process, the ATP is going to be formed by the substrate level phosphorylation only at the steps of 1,3-biphosphoglycerate and phosphoenol pyruvate. Second question. Which of the following enzyme does not take part in TCA cycle? So for this you should know the metabolisms perfectly. Options are A. Pyruvate dehydrogenase, B. Citrate synthase, C. Isocitrate dehydrogenase, D. Malate dehydrogenase. The citrate synthase, isocitrate dehydrogenase and malate dehydrogenase all are going to be present in the TCA cycle. But the pyruvate dehydrogenase is going to be the one which is involved in the connection of pyruvate converting into the acetyl CoA. So that is not going to be the part of the TCA cycle. So obviously pyruvate dehydrogenase is the enzyme which is not involved or does not take part in the TCA cycle or citric acid cycle. Then moving to the third question. The reverse transcriptase used in genetic engineering was discovered by options are A. Temin and Baltimore, B. Temin and Arbor, C. Smith and Baltimore, D. Smith and Arbor. So here they are asking about the reverse transcriptase, not about the restriction endonucleases. So many of you may get confused that you will think that restriction endonuclease because you will read often that one, but you won't read this reverse transcriptase enzyme discovery. So there you may lose the marks. So read it very clearly. So here the answer is going to be the Temin and Baltimore. So this Temin and Baltimore are going to be the one who discovered the reverse transcriptase, okay, in the year 1970, and both of them share the Nobel Prize for the discovery of this one. And then coming to the Arbor, that is Mr. Werner Arbor, who is a Swiss microbiologist and genetist, and he also worked on the enzyme called as restriction endonucleases with other scientists like Hamilton Smith as well as the Daniel Nath. So Arbor and Hamilton are related to the restriction endonuclease enzyme. Okay, so you have to remember. So the other combinations are wrong combinations. So here you have to remember the Smith and the Arbor are related to the restriction endonucleases. So their work led to the development of the rDNA technology in a rapid manner. Then fourth, the most widely accepted mechanism for DNA replication is options are A. Conservative, B. Dispersive, C. Semi-conservative, D. Evolutionary. So in this picture you can see the conservative, semi-conservative and dispersive mode, and we all well know that the most accepted mechanism is nothing but the semi-conservative mode of replication, where one strand will be from parent and one strand will be the daughter or the newly synthesized one. So that's why this is called as semi-conservative mode of replication. Fifth question. The metabolic pathway common to both fermentation and respiration is options are A. Glycolysis, B. Synthesis of acetyl CoA from pyruvate, C. Electron transport chain, D. TCA cycle. So we know that the glycolysis is going to occur both in the aerobic and anaerobic conditions. So obviously the glycolysis is going to be the answer, which is a direct question. Then sixth, clenofragments. Options are A. Phosphorylates 5' ends of DNA molecules, ligates the DNA molecules or fragments, C has endonuclease activity. D has 5' to 3' DNA polymerase activity. So clenofragment is going to be a large segment of DNA polymerase enzyme. Okay, and this is going to have the 5' to 3' DNA polymerase activity. The rest of the things are going to be absent. Why? Because so here if you observe, the clenofragment is going to be of derived from an organism called as E. coli DNA polymerase 1, which retains the polymerization from 3' to 5' exonuclease activity, but has lost the 5' to 3' exonuclease activity. And this is going to have the polymerization fidelity. What is meant by polymerization? The polymerase activity of the holoenzyme without degrading the 5' terminus. So that's how it is going to have the 5' to 3' DNA polymerase activity is going to be retained, same like the polymerase 1 enzyme, that is DNA polymerase 1 enzyme. Seventh, which of the following is an essential amino acid? Options are A. tryptophan, methionine, C. lysine, D. both 1 and 2. So tryptophan and methionine both are going to be the essential amino acids. So we know that what is meant by essential amino acids, which cannot be made by the body, are called as essential, and they must come from the food that we are intaking. So overall we are going to have the 9 essential amino acids out of 20. So here you can see the histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine are going to be called as essential amino acid. In the examination, instead of the tryptophan and the methionine, they may ask you the other amino acids also. 8th, how many molecules of ATP are synthesized for NADH oxidation? Options are A1, B2, C3, D4. So this is also a direct question. From one NADH molecule of oxidation, we will get the 3 ATP molecules. For one FADH2, we are going to get the 2 ATP molecules. So here it's not 22, it's a FADH2. By mistake it was like that. So one FADH2 molecule is going to give rise to the 2 ATP molecules, and one GTP is equal to 1 ATP. 9th question. Dihydrouridine is a. Options are A. modified amino acid found in nuclear membrane. B. purine nucleoside found in DNA. C. pyrimidine nucleoside found in tRNA. D. it is going to be of conjugated lipid found in plasma membrane. The first and the last options are wrong. So it is going to be of a purine found in DNA or pyrimidine found in tRNA. Dihydrouridine is going to be of a pyrimid nucleoside found in the tRNA molecule. So this simply called as DHU or UH2 is going to be a nucleoside where it is going to be because of 2 hydrogen atoms of uridine is going to be joined. That means the nucleoside is going to be the result of adding 2 hydrogen atoms of a uridine and making it to be a fully that means saturated pyrimidine ring with no remaining double bonds. So that is how the dihydrouridine is highly conserved modified base found in the tRNAs from all domains of life. 10th, the 3 steps of citric acid cycle in the order of their occurrence are. Options are A. condensation, reduction, regeneration. B. condensation, oxidation, regeneration. C. oxidation, regeneration, reduction. D. reduction, regeneration and oxidation. So here the correct option is condensation, oxidation, regeneration. So here in the TCA cycle chart, you can see that you are going to have the first step of so here you can see this acetyl-CoA, that is oxaloacetic acetate or acetic acid is going to condense with the acetyl-CoA. So this step is going to be considered as condensation. After condensation, you are going to have the oxidative step. So these are all the oxidation steps. So after the oxidation step, again you are going to have the regeneration of this oxalate state. So this is going to be the, so that's how we can say that it is of having the condensation, oxidation and regeneration step. Then coming to the 11th question. In the lac operon, the absence of Y-cystone results in the non-synthesis of. Options are A. Beta-galactosidase, B. Trans-estalase, C. Permease, D. RNA polymerase. So for this we should know the lac operon unit or genome, then we will come to know what is it. So here the answer is going to be the permease. So there are three enzymes for lactose metabolism are grouped in the lac operon, that is LAC-Z, LAC-Y and LAC-A. The LAC-Z is going to encode an enzyme called as a beta-galactosidase, which is going to digest the lactose into two constituent sugars called as glucose and galactose. Then coming to the LAC-Y is a permease. The question that was asked is a permease that helps to transfer the lactose into the cell. Then coming to the LAC-A, the third gene is a trans-acetylase, the relevance of which in lactose metabolism is not known clearly until now. So the single mRNA transcript is going to include all these three enzymes, that is galactosidase enzyme, permease enzyme, then trans-acetylase enzyme, coding sequences, and this is going to be called as a polycistronic. So if an mRNA is going to have all these three enzyme coding sequences, then we call such type of mRNA as a polycistronic. And a cistone is going to be equivalent to a gene. 12th question. The PCR technique, that is polymerase chain reaction technique, was developed by. Options are A. Kohler, B. Milstein, C. Kary-Mullis, D. Altman. So we know that Kohler and Milstein are involved in the hybridomer technology, that is the production of monoclonal antibodies, for which both of them shared the Nobel Prize in the medical and physiology aspect. Then coming to the Altman, so Altman worked on the catalytic properties of RNA, so where rDNA technology has developed. So only one left, that is Mr. Kary-Mullis. So this Kary-Mullis developed this PCR technique in the year 1983, who is an American biochemist, and he also won the Nobel Prize in the field of chemistry in the 1993 for his invention of developing this PCR technique. 13th, DNA staining is done by. Options are A. Giemsa staining, B. methylene blue staining, C. crystal violet, D. Fluorescein staining. So the last one is going to be the right option for this. DNA staining is done by Fluorescein staining. So this is going to be discovered by Robert Fluorescein. It was named as Fluorescein. And this Fluorescein staining is mainly used in the histology aspects to identify the chromosomal material or the DNA in cell specimens. So it is darkly stained. That means when it is going to be stained, the DNA is going to be darkly stained. And this is also the staining is also going to depend on acid hydrolysis of DNA. Therefore, fixative agents using strong acids should be avoided. Then 14th, beta pleated sheets are examples of proteins. Options are A. primary structure, B. secondary structure, C. tertiary structure, D. quaternary structure. So obviously the beta pleated sheets and the alpha helix structures are involved in the secondary structure of proteins. Then what about the primary structure? The primary structure is going to have only the amino acid sequences, and the tertiary structures are going to have the overall 3D folding driven by large interactions between the R groups. And the quaternary structure is going to be the association of several protein chains or subunits. That means each subunit is going to consist of primary, secondary and tertiary structures. So the detailed structure of primary structure I have explained in one of my video, that is protein structure. There you can go through it to understand more. Okay. So this is what again I want to just keep a note on the secondary structure. Then 15th, Which site of the tRNA molecule binds to mRNA molecule? So this is also a direct question. Is it a codon, anticodon, C, amino acid, D prime, 5 prime end? So which site of tRNA molecule binds to the mRNA molecule? So that is nothing but the anticodon. 16th. Reactions that replace cycle intermediates are called. Reactions that replace the intermediates are called anaplerotic reactions, B. Amphobilic reactions, C. Cyclic intermediates, D. None of the above. So obviously it's a direct question. It's a anaplerotic reaction. Then the mechanism or process is called as anaplerosis. 70. The two strands of the DNA double helix are held together by. Options are A. Hydrogen bonds, B. C double bond, C double bonds, then hydrophobic bonds, D. Peptide bonds. And the right answer that we know is hydrogen bonds. So these hydrogen bonds are going to occur between the two strands and involve a base from one strand with the base from the second in the complementary pairing. So these hydrogen bonds individually are so weak, but collectively they are going to give the quite strongness. That's the reason why we will write the adenine is going to pair with thymine and you will keep the two double bonds there. So those are hydrogen bonds. Then again, guanine is going to have the three bonds between the cytosine and the guanine, and those three bonds are also the hydrogen bonds only. 18. Who proved that DNA is a genetic material? Options are A. Avery, MacLeod and McCarthy, then Jacob and Monod, Biddle and Tatum, and Ivanovsky. So this is also a direct question. So initially the Griffith started off proving the DNA as a genetic material, but finally it was proved by Mr. Avery, MacLeod and McCarthy. Then moving to the 19th question, oxidation of which substance in the body yields the most calories? Options are A. Glucose, B. Glycogen, C. Protein, D. Lipids. So we know that the lipids are going to be the high enriched food. When it was oxidized or broke, then we are going to have the high release of high energy. So fats or lipids are going to have the highest energy content of oxidation of about 9 kilocalories per gram, while the proteins and carbohydrates are going to release only 4 kilocalories per gram of energy. Then 20th, Krebs cycle starts with the formation of a 6-carbon compound by chemical reaction between. Options are A. Malic acid and acetyl-CoA, fumaric acid and acetyl-CoA, oxaloacetic acid and acetyl-CoA, succinic acid and acetyl-CoA. So this is going to have the direct question. So we should know the TCA cycle in detail such that if you are getting question in any manner, then obviously you can answer. So here the answer is going to be the condensation step you are going to write, that is oxaloacetic acid and acetyl-CoA combined together, and they are going to give rise to the 6-carbon compound, that is citrate or citric acid. Then 21st, promoter is. Options are A. Upstream RNA sequence of an mRNA which recognized by translation initiation factors in order to initiate translation. Then second option, upstream DNA sequence of a gene which is recognized by DNA polymerase in order to initiate the transcription. C. Sequence of amino acid in a protein which promote catalysis of an enzyme. D. Sequence of amino acids in a protein which specially or specifically promote oxidative or reductive reaction. So promoter, they are asking whether it is going to be of located in which. So it is not mRNA, that is it is not going to present an mRNA, so that is not going to be the correct. So here it is going to be called as a location where the RNA polymerase can bind and starts off the transcription. So obviously here you can have the answer as upstream DNA sequence, present in the upstream of DNA sequence which is recognized by RNA polymerase in order to initiate the transcription. That is nothing but the mRNA synthesis. So that's the same thing, I have made a note here. Then 22. Nucleoside is a nucleotide minus sugar group, nucleoside minus nitrogen base, nucleotide minus phosphate group, nucleoside minus sugar and phosphate group. So this is also a direct question. Nucleoside means side, you are going to have the two sides. Remember, S indicates the square, that means 2. So what are those two that are going to possess is going to be of, except the phosphate, other two are going to be present. What is it? So you can see here the note, that is going to be of your nitrogen bases and the sugar moiety is present, then we call it as a nucleoside. Along with it, if you are going to have the phosphate group, then we call it as a tide. Tide means 3. In that tide, you are going to have the T letter. So there you can remember it as a 3. So this is a very small tricky, you can remember very easily. Side, it poses 2. What is absent here? Phosphate is absent. In the tide, phosphate group is present. 23rd, the RNA polymerase holoenzyme specificity factor that mediates the promoter recognition in E. coli is. Options are A. Delta subunit, B. Alpha subunit, C. Sigma subunit, D. Rho subunit. So here it is also direct. So these are all the subunits that are going to be present in the RNA polymerase, that is delta subunit, alpha subunit, sigma subunit. They are asking the initiation factor. So the rho is not the initiation factor, so it is a termination factor. So remove the rest of the 3, delta, alpha and sigma. The sigma is the right answer. So here the sigma factor is going to bind to the RNA polymerase to promote a sites within the DNA sequences to allow for initiation of the transcription, that is synthesis of mRNA. And these sigma factors are going to be specific for gene and are affected by the cellular environment, and this is going to regulate at both the transcription and the translation level. So if you check the RNA polymerase enzyme, it is going to have the 2 alpha subunits, 1 beta subunit and 1 beta prime subunit, okay, and a sigma factor. And the sigma factor along with this 2 alpha, 1 beta prime is going to be considered as a holoenzyme, which is essential to initiate the transcription at the target promoter site. Then 24, Ames test is a test that uses. Options are A special Salmonella strain to test chemicals for mutagenicity and potential carcinogenicity. 2. A Streptococcus strain to test its pathogenicity on humans. A Calyobacter strain to test for use in the treatment of mutagens and carcinogens. D. A Helicobacter strain to test for curing the gut cancer. So this is also a direct question. This is mainly used to identify the mutations. So the thing that we can directly answer is a special Salmonella strain to test the chemicals for mutagenicity and potential carcinogenicity is nothing but done by the Ames test. Dash is the transfer of DNA from a donor to recipient bacterial cell by a bacteriophage. So this is also a very easy question. So options are transformation, conjugation, translation and transduction. So they are asking the transfer of the genetic material from one bacteria to the other by means of a bacteriophage. So that is virus. So here the answer is going to be the transduction. If it is a case of transformation, it is going to be a naked DNA is going to be transmitted between the donor and the recipient. Conjugation is going to be a means of conjugation tube formation, that is a contact. So that is also wrong. Then what is a translation? The translation is a process of synthesis of protein synthesis. Okay, so this is all about the 13 series. So in the next series, we will go with 25 more important questions. Thank you.